and I'm going to be recording this so that I can post it later as a video in case you missed something or you want to go back over it. Or you just want to hear my voice for some odd reason that I would not want to know about. Alright, so some of these, like the pea plants and Mendel's laws, we're going to go through kind of quickly. I don't need to teach you genetics. You should have gone over all of that in 7th grade. So I'm not going to spend a lot of time on it now. But there's some background stuff we need to know. Now, inherit and genetics, you need to know what those words mean, but you should have them in your vocabulary list that you started Friday after the quiz. But I'll give you a few seconds if you want to write those down. And Mason is here. All right, we good? Give me a thumbs down if you're not good. All right. So we're going to move on. Inherit just means that you're getting it from your parents or the ones that came before you. Like you inherited money from your parents, or you inherited a piece of land from your Uncle Bob. Okay? In this particular sense, though, we're talking about genetics and the genes you inherit. And then genetics is the study of gene inheritance. All right, so this is a short video. I think it's 3 minutes. We're gonna try to play it. The link is up there. So if you want to jot that down, or you can get it from the video later if this doesn't play too well for you. It is a TED Ed, so you could also go to TED Ed and search for Mendel's pea plants. But we'll see how this goes.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous P mom will give each P kid a yellow dominant allele, and the green homozygous P dad will give a green recessive allele. So all the P kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
Alright. So, hopefully, what are you doing, Ted Ed? Come on, thank you. Alright. Now, I'm going to try something here. I don't know. Aha! Alright, so on your screen, you should see the questions on the left, and you should see a Classcraft team on the right. So I need Mason, Layla, London, or Mikey to unmute yourself and tell me the different variations of traits are called blank. What do we call the different variations of a trait?
Is it allies?
Alleles, but yes, it's pronounced alleles. All right, so let's do a, we'll do a quick reward. All right, answer the question. 25, 5 is what I usually do. All right, and then that goes to the whole team, I believe. All right, next team. So Micah, Alexandria, Janelle, or Britton, I need to know if you have two of the same allele, what are you?
Homozygous.
Okay.
Homozygous.
Excellent. Micah and Janelle both. All right, answer the question. 25. I need to set this as a regular thing. All right. Next question. If you have two different alleles, then you are. I need Lauren, Jan, or Sasha.
Heterozygous.
Heterozygous.
Yep. All right, Jan and Lauren. I'm sure the rest of you knew it. They're just quicker with the buzzer. All right. Whoops. And next, Bennett, Jaden, or Ramya. The genes you have is your blank.
Hey guys, would that be the genotype?
That would be the genotype.
Okay.
So if you're talking about the genes, it's the genotype. Okay. Whoops. Now, I admittedly have not put anything in Classcraft this week or last week, I think, and I need to fix that. But you can still log in and customize your character. That's what the goal is for. All right, and lastly, Micah, Alex, Janelle, or Britton, the traits that are actually seen or detected, what are they called?
The phenotype.
We got it in the studio audience. How about the at-home audience? You guys.
A phenotype?
It would be the phenotype. The way I always remember that is genotype is your genes, genes,
Phenotype is
geno,
Physical.
and phenotype is the physical, so they both start with PH. Alright, so let's move on. So those are the answers there. Alright, Mendel's laws. Yeah, I would write these down. There's more to it if you read the section, but those are the things I need you to know for 8th grade science. For every trait, there's at least 2 alleles. Some things there's more than 2, but there's at least 2 alleles. One of them is usually dominant, and at least one is usually recessive. The dominant trait is called dominant because it dominates or hides the recessive trait. So the only way to have the recessive trait is to have 2 copies of it. You have 2 copies of every gene. You have one from mom and one from dad. Oh, Janet's here. And Janelle's here. All right, we have perfect attendance for the A-Day 3rd period group.
Oh, you can move on.
Oh, okay. Are we good at home? All right. I saw a wave. I'm gonna assume the wave means I need another minute or a few more seconds. Let's see here. This is annoying. How I go to there, and how small? What? Technology is great when it does what it's supposed to do. And all the other time, it's annoying. About as small as I can make that, I think. Alright, now I got to find this. Bring that back up. Alright, we good? Alright, okay. We're gonna skip this one. So P color is a trait, okay? Tallness, leafiness, having leaves that are parallel, having leaves that alternate, okay? These are all traits. Green pea versus yellow pea would be a what? Anyone?
Trait, like.
Well, pea color is a trait. Green or yellow are different versions of the trait. So what do we call the different versions of a trait?
So they're the alleles.
That's the allele. All right. So the different forms of the trait are alleles. And then if you have 2 dominant, you're a homozygous dominant. If you have 2 recessive, you're a homozygous recessive. And if you have 1 dominant and 1 recessive, you are heterozygous. Homo, of course, means the same. Hetero means different. Zygos is like zygote. It means joined. So homozygous means you have two alleles that have come together and they're the same. Heterozygous means you have two alleles that have come together but they're different. All right. So these, just combined some questions there for the review. You should do the reviews listed in the Google Classroom assignment or on the plan. If you're ever not sure what to do, look at the plan. All right, 4.4 DNA. This is one you need to copy down. And it's whoops. Nope, that's it. Okay. So, kind of a lot. I'm going to go over 3 minutes. Oh, oops. There's nowhere I can put this. That's it. All right, so I'm going to start it and then minimize it. Yeah, if you need to move your desk to the side or something, feel free to. Oops. That's weird. Mm. I noticed some of you were using the emoticon thumbs up. I appreciate that. And of course I appreciate the thumbs up and whatnot from the people with their video on.
How do you pronounce that?
Pronounce what?
What letter A is?
Deoxyribonucleic acid.
Deoxyribonucleic.
Mm-hmm.
That's so extra. Why are such sciencey words so extra?
But it's not extra. Every part of that tells you something about it.
I know, but like, why does it have to be so long just to tell you?
You hate it?
No, I'm just wondering, like, why are such
Because
scientific
the
words?
prefixes and suffixes tell you about the thing. If you learn Latin and Greek prefixes and suffixes, your life becomes so much simpler.
So, will you?
You okay? Alright, so let's see. Oh, time is up. I thought it would make a ding or something. I guess not. I can award points. That's a thing? So let's see, I guess I would do that. And then I would be like, It's interesting. Very interesting. Oops. I'm gonna give you 5 there. I'll give you 25 there. Haha. Everybody gets some experience points. Alright, so DNA is the genetic material. It makes up the chromosome. All the chromosome is there's a protein, and then the DNA wraps around it and 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 around it. And then there's multiple proteins, and that just keeps wrapping and supercoiling. So that's what a chromosome is. The reason chromosomes form is so that the DNA is going to replicate itself. Now you have 2 copies of the DNA, and then it's going to supercoil, and then one goes to each new cell when the cell divides. And then it uncoils and becomes, DNA again, or regular DNA. It's the instructions, it's the blueprint, it's, how the cell knows what to do. Let's see. Kind of looks like I have a halo. It's weird. All right, anyway. DNA stands for deoxyribonucleic acid. You should have heard that before now. It's long chains of nucleotides. You should have known that from chapter 2. And it's a double-stranded double helix. So you have 2 strands, and they wrap around each other so that they form like a spiral staircase. In DNA, there are 4 bases. Adenine, thymine, cytosine, guanine. A always bonds to T, and C always bonds to G. In RNA, the thymine gets replaced with uracil, but more on that later. All right, so we good here? Okay. All right, just for practice, when the complementary strand for this DNA, I got a typo I got to fix there. All right, so the G bonds with the C. Guanine and cytosine always bond together in DNA. Adenine always bonds to thymine, thymine to adenine, so forth and so on. All right, with RNA, like I said, all the bases are the same except thymine becomes uracil. Okay, doesn't really matter why right now. I honestly don't know why. It's just the way the universe made it. All right, so guanine still bonds to cytosine. Adenine is going to bond to uracil. The thymine in the DNA will still bond to an adenine in the RNA. All right. Yes, this is the one I watched earlier. All right, so we're going to try. How did the last video work for most people? All right, I got a couple of thumbs up. I don't see any thumbs down. I do see, okay. Well, again, the link is there. So what you can do is go to YouTube. If you search Teacher's Pet DNA Structure, you should be able to find this video to watch afterwards. If the sound is going in and out and it's annoying, you can mute yourself for a few minutes. Or mute your computer.
Requires some form of All cells require some form of instructions to be able to function properly. They need guidelines, rules, codes for making materials in the cell. And that code is DNA. DNA stands for deoxyribonucleic acid. D for the name of the sugar, N and A for nucleic acid. DNA contains the information that determines inherited characteristics. It has the code for making proteins. DNA is found in the nucleus of eukaryotic cells and in the cytosol of prokaryotes. If we take a closer look at the chromatin inside the nucleus, we can see the structure of the DNA. The DNA has repeating subunits, and those subunits are called monomers, or nucleotides specifically. The nucleotide has 3 main parts: a phosphate group, a sugar, and a nitrogen base. In DNA, the name of the sugar is deoxyribose, which is part of DNA's name, and there are 4 nitrogen bases in DNA. The bases are thymine, adenine, cytosine, and guanine. 2 of the bases are purines, which have a 2-ring structure.
and 2 bases are pyrimidines, which have a 1-ring structure. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. I remember the pyrimidines are the bases with a Y in their name, just like pyrimidine has a Y in its name. A purine always pairs with a pyrimidine, and the slanted shape of the DNA molecule causes it to form a spiral or helix. Because DNA is double-stranded, we use the phrase double helix to describe its structure. There are 4 scientists who are credited with discovering the shape of DNA, and they are Watson, Crick, Wilkins, and Franklin. All of them received a Nobel Prize for their work, except for Rosalind Franklin. She died before the prize was given.
All right, so I'm gonna throw something out here. What happened is, I believe it was Watson, the guy there on the left, and him and Crick had been working on the structure of DNA for a while. Watson hears that Rosalind Franklin has been taking X-ray photographs of DNA, also trying to figure out its structure. And he goes over to talk to her about it, and he sees some of her photographs while he's waiting in her office. He then has a moment of enlightenment. It snaps, and he runs out, goes to the pub where Crick was hanging out, and starts talking about it with Crick. They then publish a paper and get most of the notoriety and credit. But neither of them would have gotten there, certainly not when they did, without Rosalind Franklin's work. The reason she did not get, Watson and Crick got Nobel Prizes. Rosalind did not, because unfortunately she died before the Nobel Prize was awarded. So I always like to point that out, because Watson and Crick would not have got where they got. They would not be known today. And a lot of textbooks, especially older textbooks, only mention Watson and Crick, and they leave Rosalind Franklin out completely.
Wait,
So-
what does Wilkins have to do with the discovery?
I'm not sure what Wilkins did. I'd have to look Wilkins up.
Did he like work with her or something?
I don't know.
Oh.
Like I said, I have to look it up.
Conspiracy theory,
Alright.
they killed him,
Each of these scientists played a role in piecing together the structure of DNA. They learned that along the sides of the molecule was a backbone made of alternating sugar and phosphate molecules. On the inside, like the rungs of a ladder, are the nitrogen bases. Adenine and thymine form hydrogen bonds together. Cytosine and guanine form hydrogen bonds together. To help me remember which bases link together, I think of writing the letters. A and T both use straight lines. C and G use curved lines. I also know that A and T have 2 hydrogen bonds, but C and G have 3 hydrogen bonds by saying AT2, CG3. Silly things like
All right, so I'm gonna stop there because that's all we really need to know. All right. So where's I need my random picker again. All right. Wheel of Destiny. We'll do a player this time. Moo ha 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 ha. Come on, there we go. All right, Britain. DNA is the blank for cells. You might have to unmute yourself.
blueprint.
Very good. Okay, so they are the blueprints. Whoa, not 50. Okay. All right, and let's see what's next. Janelle, DNA is made of monomers called?
It's the 4, right?
No, those are the bases.
Those are the-
What are the building blocks of DNA? It's made of a phosphate, a sugar, and the base.
Oh.
Look in your notes. DNA is made of a long chain of.
Oh, I didn't get to that one. You switched to the next slide before I got there.
All right, Jan, you help her out.
Number 2, is it nucleotides?
Yeah, it'd be the nucleotide.
Oh.
All right.
Can I see that? I need to write that down.
Okay. So DNA is made of monomers called nucleotides. All right. Let's
I don't
try
have it written.
Why don't you have it written?
another. London. ACTG are the 4 blanks in DNA. Right. They would be the bases. Oh, there you are. All right. Okay. Let's see who's next. Layla Andrews. DNA is a blank blank shape.
Um, uh, is it?
Look back in your notes if you need to.
The double hexyl
Helix.
helix.
Helix. Double helix. Excellent. Alright. Alright. And let's see here. Micah, what does A bond to? You might need to unmute yourself.
The T.
Right, A bonds to T, which means, of course, that T bonds to A. All right, so number 7, Sasha, what does C bond to?
G.
Very good. A to T and C to G. G to C and T to A.
That was confusing for me.
All right. So there are the answers for anybody watching the video that needs to check those. 4.4 review. We're gonna skip that video. Again, DNA is a blueprint. DNA makes RNA, and the RNA makes proteins. All right, structure and reapplication. I would write the heading. You always write the heading. Otherwise, how do you know what the notes are about? So you always write the heading, and then I would write number 2. Okay, so just number 2. Heading number 2. Skip number 1. Okay. So let's see if I can pull up my. See, I need a third screen. That's what I need. I need a third screen. Normally I'd use my phone, but it is busy being a camera. Oh, goodness. So time's up. Are we good? Do we need another minute?
You said only the first one, right?
Number 2. The heading and number 2. All right, so DNA structural replication. DNA is a double helix, as we said, so that means it's 2 strands. So an enzyme has to unzip those strands, and then another enzyme, take this strand, another enzyme will take a nucleotide and match it up with whatever the base is. So this is an A, it puts a T there. And then it keeps doing that one at a time. And it builds the new strand using the old strand as a template. And then over here on the other side, the same thing is happening, but it's happening in the reverse direction. Okay? So each new molecule of DNA will use one of the old strands and have a new strand that goes with it, which is kind of cool. All right. So we good? We need a minute? Okay. I'm actually going to skip this one. I feel like it got a little more complicated than need be. But, well, that gets in RNA. All right, I'm going to go back to it. Again, the link is up there if you want to get that, and you might be able to play it through YouTube yourself easier than watching it here. I don't know.
DNA. We talk about it so much, it's the ultimate director for cells, and it codes for all your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where. Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when. When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has 2 strands, you can think of helicase unzipping the 2 strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3'. And the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase, once again, is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors? In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA basis, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions in the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
What happens if it makes an error?
If there's an error in the DNA copying,
Yeah.
then that's a mutation, and mutations are coming up. We're gonna talk about those in just a second. Alright, I'm gonna start my video back. Alright, so, now a lot of that is stuff you don't need right now, but, it's not gonna hurt you to have it in your brain rolling around when you get to high school. Alright, cells make a copy of DNA when they go through blank and blank. When they go through what? Alright, DNA replication, but why would they need to copy their DNA at all? They already have their DNA.
Because cell division can't happen without it.
Right, cell division. Alright, words ending in ace are usually blank,
And
usually enzymes. That's one to stick in your brain and keep there. If it ends in OSE, it's probably a sugar like ribose, deoxyribose, glucose, sucrose, fructose. If it ends in ASE, it's probably an enzyme. All right, this one, just to give you a vague picture, okay, of protein synthesis. You have the DNA in the nucleus. It makes a strand of DNA, or I'm sorry, a strand of RNA. That's the messenger RNA. And it goes floating outside of the cell, or outside of the nucleus, into the cytoplasm, right? And it meets up with another piece of RNA called rRNA, or ribosomal RNA. And that ribosomal RNA and the mRNA join together, and then a piece of tRNA, we call transfer RNA, it brings amino acids one at a time to build the new protein. What I'm going to ask you to write down, though, is this chart down here, so that we know the differences between RNA and DNA. Whoops, my bad. So I would just copy this chart. All right, so let's see here. Let's go back to my timers. And start a timer. All right, I'm going to give you 2 minutes. Start.
Hello.
What's up?
I need your keys and your credit card. Don't ask questions. Hello, students. Thank you. Bye, students. Bye, students.
All right, I have one more slide after this one. All right, you have about 20 more seconds, so if you need longer, give me a wave or something. All right. I'm going to skip that. It's not a bad video if you want to look it up, and I'm going to skip that one as well, and skip that. I'm even going to skip that because I just want to get here. All right, These you need, all of these. So, a mutation is a permanent change in the sequence of DNA. Once the mutation occurs, every single copy of that DNA strand is going to have the mutation. Most mutations, they happen all the time. I mean, you're literally made of trillions of cells. All those cells have to make copies of their DNA at some point. They happen all the time. Most of the time, they get fixed by DNA polymerase. Other times, they have no effect. Most of the time, they have no effect. Sometimes they can be harmful, and sometimes they can be beneficial, but sometimes they're not beneficial until combined with other mutations over a very long period of time. Okay?
For example. What is it?
For example, the changes in tortoise shells. If you look at the tortoises of Galapagos Islands, Darwin noticed that the natives could tell what island a tortoise came from by the shape of its shell. There are some tortoises that had a high neck in the shell so that the tortoise could lift its head up to eat fruit from plants or whatever. There were other tortoises that had different shaped shells for different reasons. But those changes didn't happen overnight. It's not like Billy the tortoise had a mutation and all of a sudden there's a new species. There was little mutations that probably had no effect whatsoever until there were other mutations, and then all the mutations together led to a bigger change. They can occur in the DNA itself, or a whole chromosome can mutate. Remember, you have a total of 46 chromosomes. You got 23 from mom, 23 from dad. And sometimes the mutation can happen in the chromosome itself. If you think about it, chromosome is your DNA super coiled into a tight little package. And think of like you take a rubber band and you twist it, and you keep twisting it, and then it just starts twisting around on itself, and then the twisting turns around itself, and it just gets into a smaller package. That's what DNA does when it becomes a chromosome. So if part of that chromosome gets deleted or flipped around or something, that could be hundreds of genes that are affected. So it's a big deal. I mean, if you took the first Harry Potter book and you went in and you changed one word, in the whole book you changed one word, it's probably not going to matter a whole lot. You change 1,000 words in the Harry Potter book, even if you change them randomly, it's probably gonna have an effect, okay? But what if you took the book and you just ripped out like the last 30 pages? Or you ripped out 30 pages in the middle of the book?
Then the rest isn't gonna make sense.
Right. So then, you have problems. Mutagens are chemicals or types of energy that cause mutations. UV radiation is a mutagen. That's why it damages your cells, because it damages the DNA in your skin cells. That's why it increases the chance of skin cancer. So wear sunblock. Mutagens are substances that increase the chances of mutation to occur. Yeah. And they're far more likely to cause cancer than to give you superpowers. Alright.
I have a question.
Yes.
So let's say, how long would it take for the cell to replicate itself? Is it, or is that not known?
Well, it depends on the cell. Some bacteria can reproduce every 20 minutes. Other cells take longer.